This is episode 89 of the Immunology Podcast, Host-Microbiota Interactions with Dr. Theresa Alenghat. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Theresa Alenghat from Cincinnati Children's Hospital Medical Center on the podcast to talk about her research on host microbiota interactions and mucosal immunity. We also got our usual roundup of recent highlights in immunology news coming up. But first,
Immunology 2025 is the world's leading annual all-immunology meeting, and this year it's in Honolulu, Hawaii. Join us from May 3rd to 7, 2025 to connect, collaborate, advance your career, and access the cutting-edge immunology research. Abstracts open for submission October 1st, 2024, and you can learn more at immunology2025.aai.org. And Jason and I will be there.
aloha.
Hey, excited.
How are you?
I'm good. You?
Good. We're recording this on Jewish New Year, right before I head to synagogue. So,
Happy New Year.
little synagogue time, little food time. It'll be good.
Oh, that's nice. Food is always good. So happy New Year 5,327, whatever that year might be.
Some big number.
Some big number. All right. Very nice. So I'm looking forward to talking to our guest today. She's a trained veterinarian, apparently.
Yeah, it's not that uncommon in the host microbiome space.
Right? Do you have pets? I don't think I've ever asked you.
No, my immune system, as we have discussed, is very aggressive, and I also have allergies to all cute, fuzzy things.
Oh.
I had a pet turtle growing up.
Turtle? A tarantula.
Oh, that will not fly in my house.
They would eat the flies.
There was a pun there.
But yeah, that's kind of sad. I'm sorry for you, Jason.
You have a cat. I see it on screen all the time.
I have two cats. It's just very similar to each other, so you might have not realized they were two different cats.
You have two black cats?
Black and white, two nice tuxedo cats. Very, very smooth, very elegant cats.
There's one named T-Reg and one named CD8 or something.
No, my boyfriend got to name the cat, so they were just going for other kinds of names. We know we are Spanish speakers, so the first cat is called Vecino. It's like neighbor. And according to my boyfriend, our second cat, which is kind of my cat, he's called number 2, but not completely agreeing with that. So I call him Segundo, which is also in Spanish a name. I mean second. So that's good. But in the end, we just call him gato, gatito, and different versions of like cat in Spanish. So very generic.
That's adorable.
They're quite adorable. They're kind of nice.
Senior Gato, Mr. Cat. Or Senior Gatito, Mr. Kitty.
Exactly. All right. Enough of my cats. So what are we up?
Oh, I have a great segue. Today? Well, you know, animals love cheese. And today I have an article for you about cheese.
All right. Sure. Do all animals love cheese? I'm not so sure.
Have you seen dogs around cheese? Oh, my God.
Oh, well, dogs will eat anything. I'm not sure my cats would eat cheese. I'm not sure. Maybe.
Just experiment.
If I allow them. All right. So what's up with cheese?
So this is just a cool, weird paper, and I love it because it's cool and weird. So it's titled, Bronze Age Cheese Reveals Human Lactobacillus Interactions Over Evolutionary History. First authors are Yi-Chen Li, Bao Mi, and Wenying Li. Last authors are Yimin Yang and Kwame Fu. Came out in Cell, 25th of September. So they found some tombs where people buried people with cheese. And so they have some like Bronze Age kefir cheese is what they found it to be. So first off, like, this is just like, hey, you found something. So what do you do with it? You sequence it. So this group found 3,500-year-old cheese, of which a lot of it was contaminated with other bacteria, but they did get signals from a lactobacillus strain that is found in kefir grain, because that's how you make kefir cheers. Is you take this grain and you mix it in with the cheese, and then the bacteria does this thing called Lactobacillus kefirano-ofasciens. Okay, kefir lacto, as one names things. So what they did is they sequenced it and they did this by using specific primers related to modern lacto kefir, as I'm going to call it, and saying the thing, L kefir, because otherwise they would have problems with read depth with all the degradation and everything. And so they were able to overcome that pretty uniquely. So that's one of the key things this paper did, was it figured out, right, we did it at full sequencing and saw it was there. Let's do a deep sequencing with these specific strains, with the specific, keyed primers, to make sure you can get the whole library. Because they did whole genome sequencing and they wanted to understand how it genetically drifted over time. And so this is where it gets into like, well, what does it tell us about people too? And that's what's cool. So there's two strains. There's this Kefirano-ofasciens and the Kefir granum. What they say is that they see the spread of kefir from this, the wheat that these bacteria grow on, right? The cheese. And they say that this granum clade is mostly in Europe and Eastern Asian coastal and inland regions. That shows that it spread from the Caucasus region to the rest of Europe and the coastal regions and islands of East Asia. Whereas the other one, the Kefir-ofasciens one, has a separate route from Xinjiang to inland East Asia. And so they see, actually, because of their ability to sequence this, compare it to modern day, and they're able to track it. And then the other thing that they're able to see is that there's certain genes that appear in modern strains, like antibiotic resistance and ability to tolerate various environments. And they can actually see these genes pick up over time by comparing to other samples in modern day. They're adapting to humans and the human immune system and where we have gone based on when these genes appeared and where. So you don't have antibiotic resistance until modern times, right? But your ability to tolerate different climates are somewhat for bacteria based on the genes too. And they see these tolerance genes pick up with the migration. And so it shows that humans from one area migrated to another area in two routes over time, bringing kefir with them and causing the bacteria in them to adapt. And it also, interestingly, and this is the other part of human immunity that gets into, is that people in this area are lactose intolerant. But this bacteria chews up most of the lactose in the milk and making cheese. And so you see that functionality be preserved in the lactose intolerant populations. But if you don't need that, then, in a lactose tolerant population, you have less of that gene expression. And so they're actually able to, it's just interesting because from this thousands of year old cheese, they can track migration of food, grains, and people all together. And then they also see the goat DNA in the dairy because it's goat cheese, and they can use that to show how the goats moved. So it's not super immunology, but it does have that comparative historical biology to it. And like the fact that you can, we're talking to a host microbiome person today, and just seeing this tight symbiosis has existed forever and ever. And it's just part of human history, is what we do with our bugs. We're just here to carry them, basically.
That's quite a nihilistic way of looking at it. You're just, containers for the bacteria that populate this earth. I find kind of interesting what the point that you make, that this kefir bacteria had they degraded more of the lactose and therefore would make it more appealing to populations that have lower lactose tolerance. That's kind of interesting.
The whole thing is this, it's very interesting how biology has gone on a journey with us.
Yeah, for sure. And what exactly is that they are sequencing to in order to make this phylogenetic comparisons between the bacteria?
Lactobacillus and then the substrains that are of kefir, like modern day kefir exists. So they just use the modern day bacteria and compare it to the ancient one.
Ah, they're gonna like align again. So you have like a full. Is it a full chromosome or it's just like the ribose whole genome sequence?
Whole genome sequence.
Okay, yeah, that was my question. So
That's why they had to use the deep primers.
all right, very interesting. We should have some kind of molecular archaeologist join the podcast one day and talk about
We should. That'd be really cool.
how you look. Because there's a bunch of stories lately that are really looking into human evolution through this super molecular, using these very molecular tools. And I think pretty cool. All
Yeah.
right. You know what else is pretty cool
Besides cheese.
besides cheese? I mean, it's hard to surpass cheese. I love cheese. It's transferring mitochondria from one cell to another in order to get it
Oh,
rejuvenated.
I've heard about like mitochondria moving around generally.
Yeah. I mean, that's also super weird. Why are mitochondria moving around? But apparently it's a thing. I think that it's not controversial anymore that mitochondria can move between cells. Um, what is the reason behind it? I think it's like a bit maybe more complicated, but this paper I'm going to talk about, I think shows with, convincingly that this is something that T cells can do, and that it actually might have, concrete functional advantages for T cells to be able to do this. So the paper is called Intercellular Nanotube Mediated Mitochondrial Transfer Enhances T cell Metabolic Fitness and Anti Tumor Efficacy. And the has three first authors: Jeremy Baldwin, Christoph Heuser-Loy, and Tanmoy Saha. And this is from the lab of Luca Gattinoni. Yeah, I'm familiar with some of his work, so in cellular therapy, and he's at the Leibniz Institute of Immunotherapy in Regensburg, Germany. And the paper was published in Cell online in September 13th, but I think it's technically part of the November edition of Cell. So basically, as I already kind of spoiled the beans, they see that T cells can acquire mitochondria, and in this case, they show that is from particularly bone marrow stromal cells. So it has been already established that mitochondrial transfer is a thing, and it looks like in certain cases it can aid damaged cells to survive. But I think most problematic, it has been shown to be used by tumor cells to hijack mitochondria from tumor-infiltrating lymphocytes and stromal cells in the tumor microenvironment. And there's several mechanisms by which we think this phenomena happens. But I'm going to focus about one in particular that's called tunneling nanotubes, aka TNTs. And these are basically actin-supported membrane protrusions that build a bridge between cells that are kind of separated at a certain distance. And it's become evident that there's an exchange of not only cytoplasmic factors or like soluble things, but actually full organelles such as mitochondria. So what do they do in this study? Basically, they co-culture both human and mouse. They do kind of the human and the mouse situation, uh, CD8 T cells with bone marrow stromal cells. And in initial experiments, they just put them on glass cover slip over 24 hours, and then they fix them, and they have some beautiful imaging with field emission scanning electron microscopy. So basically, electron microscopy. So there's like the first figure is this awesome pictures of these T cells kind of resting or interacting with this bone marrow stromal cells, which are fairly larger than the T cells, and they point at some clear structures connecting the surfaces of both cells. Basically, they see an average like one of these nanotubules per cell. And they see that there are this kind of filamentous structure with some kind of protrusions in the middle, like some sections that are enlarged. So when they kind of use a slightly different approach, and they have these bone marrow stromal cells that are transduced with a construct that stains mitochondria with a dye called mito-DS red, which is used often in the mitochondrial field. And they actually can see that then when they look into microscopy, they have this focal microscopy images, and they show that if they also stain for actin, that stains these filaments, they see the filaments and they see these red signals inside the filaments, suggesting that they're mitochondria inside. And then they see that if they co-culture the T cells with the stromal cells, some of this red color gets into the T cells. So they kind of really suggest that there is a transfer of mitochondria from one cell to the other. They improve a little bit their system. They have this transwell situation in which they improve the viability of the cells, allows them to do more experiments. And basically, they spend up some time showing that indeed there's a transfer of mitochondria from one cell to the other, and that these mitochondria are functional as they reach the new cell, and they contribute to the metabolic fitness of the cell they are transferred to. They show this by using measurements of mitochondrial activity, and also by if they damage the mitochondria in the bone marrow in the stromal cells before transfer. They use this ethium bromide for this. Then they see that this metabolic advantage is abrogated. So they really suggest that they really show that these mitochondria are doing something in the new cell. Um, and And I think one of the interesting things is that they look at, they try to compare what are genes that are, why are some cells taking mitochondria, where are not other cells, what are the genes that are initiating the formation of these filaments, and these nanotubes. And they focus on one particular protein called talin-2, which is a cytoskeleton protein that's involved in the assembly of the actin filaments. And that seems to be one of the key proteins involved in the generation of these nanotubes, both, and it has to be expressed in both the recipient and the donor cell. So what does this mean? You see this transfer, it affects the metabolic performance of the cells, the T cells in this case. What does it mean on a kind of a bigger picture? So they show that they have some mouse models of mice tumors. I think there are melanoma models in which they show that if they have cells that are recognizing, a tumor, if they choose, they co-culture these cells with the melanoma, with the stromal cells. They allow them to kind of take up this mitochondria, and then they kind of they sort the cells that are mitochondria positive, like they have acquired mitochondria from the stromal cells, those ones, and they transfer them in a tumor transfer experiment, the cell transfer experiment for a tumor model. They show that indeed they have a better fitness, a better performance. They are better uh, at mediating tumor regression, at infiltrating the tumors. They don't seem to proliferate more, at least when they do some measurements with the CFSE, or chartreuse violet, but they do seem to survive better, and they show less apoptosis. So basically, these cells have, they have an edge, a metabolic edge that makes them better at tumor fighting. And also, I think another interesting model they do lymphodepleted host, and they transfer the cells. And then if you have a mouse that is lymphodepleted and you transfer cells, those cells will proliferate to fill the niche that is now empty. And again, they see that there's an advantage. Cells that have this extra mitochondria have an advantage in cell divisions, and they seem to keep that increase load of mitochondria in like the daughter generations. So these kind of a long term, they keep this advantage long term. In the end, they prepare some human cells. So this previous experience were with mouse, and they have some C19 CAR cells, and they also again they show that there's an advantage in the metabolism of CAR T cells made with this that receive mitochondria from stromal cells. And I think this is probably the most interesting part is that they have they looked into TIL products, so tumor infiltrating lymphocyte products, in which you have the lymphocytes from the tumor and you expand them, in vitro. And this is usually a problem because these cells are already starting from a bad place. Cells in the tumor, they're not very happy. But they show that they can enhance the activity of the still cells by co-culturing them with bone marrow stromal cells. They have a cell line of bone marrow stromal cells, and that this increases the fitness of cells that are already to start with pretty, in a pretty bad shape. And I think this is really, really cool because I don't know if there's like people are really looking into trying to improve the fitness of these cell products because they are really good when they work. But most of the reasons they don't work is because the cells are already coming from a tumor. They're already exhausted. They're already gone through a lot. And they're hard to kind of expand in a way that they keep their anti-tumor function when they're expanding, like a million times or things like that. So I thought it's very cool paper.
So if I get this right, then some cells are just prone to this, but once you're prone to sucking in other mitochondria, you're stuck. You just have that advantage.
So once you take up those mitochondria, they become part of your cytoplasm, of your
Right.
organelle, and then the daughter cells also have an increased mitochondrial content compared to cells that did not take up the mitochondria during this transfer phase.
But what makes some cells want to take up mitochondria more than others?
Well, I think when they do this single cell, sorry, this RNA-seq, comparing mitochondria cells, like after they do the co-culture 24 hours, and then they separate cells that have taken mitochondria and they measure it with this DS red, a mitochondrial stain, versus cells that don't take up mitochondria. So they see a bunch of genes that are associated with membrane, changes in the membrane, and in particular, this one gene that I mentioned, um, Talin 2 is one of like the biggest differentially expressed genes between cells that took up mitochondria and cells that don't.
But we don't know why they chose to be that way or not, just if they are.
No, they don't look into that. At least I don't think I saw like why. It might be just stochastic. I don't know.
Okay. I'm just wondering, like, why do I choose to gobble up mitochondria?
Yeah. Yeah. Good question.
All right. So next up, I don't have a good segue here, other than it's T cells. I'm doing a CAR T paper.
Ooh.
Yeah. You have to do a microbiome paper next.
Nah, I'll leave that to you.
All right.
I go. Okay. Impress me, Jason.
Single cell CAR T atlas reveals type 2 function in 8-year leukemia remission. First author is Ziyang Bai. Corresponding authors are Li Tang, Carl June, Joseph Mellenhurst, Stefan A. Grupp, and Rong Fan. So this is looking at CAR T therapy in ALL, which has a 50% relapse rate in the first year. Not great. And so they looked at pediatric cases in 2 different clinical trials and tried to figure out what made some people responders versus not by single cell RNA seeking the pre-infusion CAR T cells and after CAR specific stimulation in them as well. So they took some of the cells that were left over, right? They sequenced them and they sequenced them after doing in vitro CAR stimulation. And they also had some healthy controls. So they identified in the end, they did single cell RNA sequencing, et cetera, and found that type 2 signaling with all this, panoply of cytokines, which I can go into in a second, was critical for this. Type 1 signaling was preserved throughout, but the more type 2 signaling you had, the better you did. And the way they did this, they had one of the trials was a single center phase 1/2, and another trial was an open label. And so they used the open label as the validation set for the training set of being the single center trial. They could see the pattern persist. And so they found that long-term responders based on single cell RNA sequencing and intracellular cytokine signaling had more type 2, including PDGR, combination, exhibition of both CD4 and CD8 combined cells, your standard cytokines, IL-3, IL-4, IL-5, IL-13, IL-31, right? And this is true again of both the CD4 and CD8 CAR cells. And that then only IL-10 wasn't really important of the type 2 cells. And so then they're like, well, that's really interesting. Can I get this to work at all in vivo or in vitro? And so they had a mouse model of human cancer, and they showed that if you take these CARs and put take a step back. They show that like, hey, these cells live longer and they don't exhaust. So they kind of look and say they don't seem to have exhaustion markers. In vitro, they just seem to persist longer when we grow them out. They hold on and stop exhausting and don't undergo apoptosis. All right. But apoptosis in exhausted cells did go up. So it killed so these cells got rid of their exhausted cells if they had more type 2 and kept everything else chugging along. And it had less markers of inhibition, the standard inhibitory signaling markers. They looked at chromatin accessibility and identified GATA3 and STAT1 for type 2, but mostly STAT1 was labeled, but GATA3 really went up, and that was a marker. But then they took this and put it in mice, and they challenged the mice with cancer. Let me find the human leukemia model, right? But then any of these high or low type 2 killed it. But when they re-challenged the same mouse with more cancer, only the CAR T high was able to do another round of killing. And so that's what they're saying. All these cells kill real quick, but only ones that are high in type 2 profile are able to do it again and again and again. And they said they challenged it with another wave and another wave. And they showed that even multiple waves in this model, the type 2 high were successfully able to do it. And then they took the type 2 low and then revitalized them by adding in IL-4. And so IL-4 switched them to a type 2 high state, and then they behave like the type 2 high cells. So now the question is, when you make these CAR T cells going forward, should you add type IL-4 to the CAR T milieu of prepping to make them more type 2 high so they have a persistent durability? And there you go.
That's kind of interesting. I think it's not that crazy that this type 2 profiles are associated with persistence. I think this has been kind of implied in other studies. Um, but I know sometimes it makes me feel because type 2 is supposed to be associated with like allergy, and so I really feel like all this. It really kind of changes the perception of what of the original significance of this type of these groups that were generated, like decades ago, and now they're like, yeah, there's so much more than allergy to type 2, quote unquote, responses and cells. Um, but yeah, I think anything that helps improve persistence, especially of CAR products, is super important. I also wonder whether the fact that these CAR T cells, especially in the case of the CD4 cells, and the idea that you have this specific direct recognition of target cells so strong. I think it's not something that you see so much on CD4 cells. I don't know if like CD4 cells, how unnatural it is for a CD4 to have this kind of really. I mean, of course there are cytotoxic CD4 cells, but I think when you think about their kind of helper function, this is a little bit it's not what they're doing right in this or do they see any kind of specialization in terms of
They see it
cytokine.
of the CD4 and 8, right? They're just seeing
Yeah.
that cells don't exhaust. If they have more type 2 high priming going in, they don't become exhausted and suppressed later on after the first round of cancer killing.
Right, because they look into the original product that the patients got.
Correct. And then they look at post-challenge too and see what happens. And they'll do CAR stimulation of the cells in vitro. But the real question is, going in, what does it look like? And the type 2 high persist in the patients, and the people associated with type 2 high have better outcomes. And what they see when they go into the mouse models is that the type 2 high can do multiple waves of killing of tumor in mouse, whereas the lows cannot.
Right.
Which is what they see in people.
And does this correlate with like the cells from the patient before the CAR product was made? Like, where is this type 2?
This is after they make the CAR cells. I'm looking at the original CAR product that they're looking at in the people. Oh, but they don't, in the people, I don't think they correlate it with the person's original milieu, right?
Right.
Because they don't know.
Right. Okay. All right. Well, thanks for sharing. So for our last discussion of the day, let's move away from all this mammalian immunology, T cells, all these
We gonna do
high
sharks?
No, think older. Think more primitive.
Bacteria?
Bacteria.
Bacterial immunology.
Bacterial
We're gonna make a
immunology.
microbiologist out of you in the end.
I know. I have to say, this is not the eukaryotic immunology podcast, right? This is immunology in general. Whatever helps you get rid of your pathogens is immunology. So that's what I'm thinking about. So, my story, I'm gonna try to be brief because there's a lot I had to learn for this study. It's called Phage Reconstitute Not Plus to Counter Bacterial Immunity. And the first author is Ilya Osterman from the lab of Rotem Sorek at the Weizmann Institute of Science in Israel. And basically, in this study, they look into bacterial immunity against phages. They're old enemies. And I think nowadays everybody thinks about bacterial immunity, and you think, well, CRISPR. That's the OG in the general public. When you think of, or general public, like general scientific public, that's like, yeah, there's adaptive immunity. That's pretty cool. But it's also pretty cool wide spread immunity that helps you against several kinds of pathogens. And I think this is where the story is going. So it turns out that if you remember from your biochem class, and you, dear listeners, remember, not H, and here I have to say, I'm not sure how to pronounce this in English. So, how do you pronounce
Nicotinamide.
Nicotinamide. That makes more sense. Like trying to pronounce, like, I'm not sure how. Okay.
Nicotinamide, adenosine. Yeah, NAD.
Dinucleotide. Yes.
Dinucleotide, yeah.
Nicotinamide, adenine,
That's NAD.
dinucleotide.
No one says NAD.
I know.
Because in English, then you start talking about your NADs, and then that doesn't go well.
Good to know. NAD then. So NAD plus, yeah, that makes also sounds better. It's critical for for biochemical processes, right? We know it's everywhere in the Krebs cycle, in the glycolysis and whatnot. To make nucleic acids, whatever you need, NAD plus. And when it's used, it's reduced to NADH, and that's kind of the reaction that happens. And unsurprisingly, bacteria really need NAD plus for their stuff, but also someone else needs NAD plus for their stuff, which is phages that infect bacteria. Because phages that infect bacteria, they need to hijack the metabolism, make more phage babies. So obviously, NAD plus is a must. So because of this is kind of an Achilles heel of a phage infection. And this is something that many bacteria will tackle if they're infected. So there's numerous bacterial defense systems that focus on depleting cellular NAD plus if they detect a phage infection. And this kind of halts metabolism of the cell, and therefore you might be able to kind of stop the phage infection on its tracks. And there are there's a list here of like proteins, enzymes that are related to this, but basically, the main action that is taken to do this is that you cleave the NAD plus, like, I don't know, in half, not really, but you cleave it, and then you have ADP ribose, also known as NAPR, and nicotinamide, and they're like separate. And in principle, you cannot kind of make NAD plus from those again. There's no known enzyme that does that reaction, so it's really kind of a one-way street. And I guess most of the cases, if you do this, the bacteria will die, but the population will survive. So that's great. So I'm good. Of course, viruses are masters at, finding a kind of counterbalancing the immune responses of the bacteria. And in this paper, they show that indeed, after some searching, they find phages that are capable of actually reversing this particular reaction that seems to be mainly aimed at surpassing at the defenses of the bacterial target. So they named this pathway that is encoded in certain phages as NARP1, and basically they show that they can rebuild NAD plus directly from this ADPR and nicotinamide, and this is something that has apparently not been seen before. So how do you go about finding a novel biochemical pathway never described before and publishing in Nature? Well, let me walk you through it. So what they show is that they start, they basically do a lot of like searching. That they search for certain proteins that are described in these phage genomes that are not sure what they're doing. They seem to be related with NAD metabolism, but they're like, well, you're obviously not making NAD from, from this ADPR and nicotinamide, because that's not possible. Nobody does that. Eventually, they do a lot of like, they find these enzymes, they express them, and they show through a lot of biochemical assays that indeed that's what those enzymes can do. They are using a completely novel pathway to regenerate the NAD plus that the phages need. And they show this by, they find certain, they do this experience in vitro, they find certain, they look at, because there's it's an operon that has two genes, and those two genes seem to act together. And they show that they find, one gene makes a intermediate form, and then with the other gene, they can have the whole pathway. And they name it NARP1. And they show that indeed by having this pathway, these phages can avoid or can get over the NAD plus depletion that happens in bacteria that have these defense systems that can deplete and degrade NAD plus. So basically, they show they have bacteria in which if these bacteria are expressing these proteins, then these phages survive. But if you remove these genes that they're studying, then phages don't survive. And what is very interesting, I think that's probably the most interesting part of why this particular system has evolved, is that because it only works with ADPR and nicotinamide. So this system is only active and kind of spending, because it does take ATP to do this reaction. So you're investing a lot of the energy available to make this NAD plus come back. And this, because ADPR and nicotinamide together are only present if the bacteria generated it from NAD plus because of their defense system, then this kind of counteracting system is only active if you have the system. You know what I mean? It's like it's only acting if you have infected a bacteria that is using NAD plus degradation as a mechanism to protect itself from you. And you're not wasting, because if you use other pathways that generate NAD plus from different substrates, you might be wasting your energy doing this when in fact it's not necessary because the bacteria did not deplete the NAD plus to start with. So I think that's kind of the point that they make. And then it's interesting because then they see that they find actually the system in other phages and they find other kinds of systems. So it turns out that, NAD plus reconstitution is more common than they initially thought. And they see these homologs in a bunch of other phage genomes. They have these databases and they kind of search for these. So I thought that was very interesting because they show a completely novel way for phages to reconstitute their NAD plus and, circumvent the bacterial defense. And in prison, I mean, it's really cool because in the end, everybody's fighting their own battle. We are fighting our battle against, the our infectious, bacteria, parasites, and cancer. And these poor bacteria, they're fighting their good fight against all those phages that are trying to, infect them and kill them.
This is super interesting. You had me at new enzyme.
Yeah. Yeah. I mean, they were pretty, I think it's pretty neat. It's pretty cool. There were a lot of like very biochemical things that kind of flew over my head a little bit, but
Didn't give me a KD, a KCAT, a Bmax. I have so many questions.
actually not that I see.
That's okay.
They do a lot of like NMR. This is
Ooh,
to look into
be still my heart.
to look into the molecules that are generated.
Oh, that's
So I think that's pretty cool.
yeah.
Mass spec, and they do a lot of things to like follow the different
Got it.
metabolites that are
They don't
generated.
profile the enzyme itself, though.
No, not that I know. They do a lot of like genetic the comparisons, and they find this enzyme in different types of phages, and they show that they're more common than you'd think.
Awesome.
But I don't see specific measures of
Okay.
like very biochemistry. Maybe in the supplementary.
Probably. All right. Well, you have me super excited. I'm super excited that we're going to be talking to Dr. Theresa Alenghat at the Cincinnati Children's Hospital in just a moment. But before we do that, I'm also super excited to talk about this one because this is how I first found about stem cell. And I'm talking to you about physiologically relevant research with Intesticult. Intesticult Media is an awesome media that provides a complete workflow for establishing, maintaining, and differentiating human intestinal organoids. You use intestinal organoid growth media to establish and maintain the organoids in a proliferative state, and then to achieve more physiologically relevant proportions of the differentiated cell types for your experiments, you can passage your culture in a Intesticult organoid differentiation media. Receive an offer to try a Intesticult in your lab by visiting us online at www.stemcell.com/try-Intesticult. I will say this stuff really, really works.
Now, for the second part of our show, for our interview, we are very happy to welcome Dr. Theresa Alenghat. She is a professor at the Immunology Division and Margaret K. Hostetter Chair at the Cincinnati Children's Hospital Medical Center at the University of Cincinnati. And the Alenghat lab investigates host-microbiota relationship and how this affects various diseases. So I think given the strong microbiota component of our conversation today, Jason's going to be taking care of most of it. But I'm very happy to have you here. Welcome, Dr. Alenghat, to the Immunology Podcast.
Thank you. It's great to be here.
Thank you for being here. All right. So where do I go that just doesn't immediately go into the weeds first? Maybe high level, could you give a little overview of how the host immune system sets the microbiome? I know we had talked about a paper where you had humanized mice and then all of a sudden their immune system looks like human or their microbiota looks like a human microbiota that just came out. But I was wondering, maybe you could start there, your little bit of how the intestinal, epithelial, MHC, the immune presentation, how you're sculpting the microbiome from there. And then maybe from there we can go into all the other really cool stuff that you've done with histones and everything.
Sure. Yeah, I guess I would say that, our focus has really been maybe a bit on the flip side of thinking about what's downstream of the microbiome. So really more of a what do microbes do to our epithelial cells or our immune cells in the gut. On the other side, the flip side of how the microbiome is shaped by the immune component, I feel like this is there's still so much that we don't know, but I think without question, if we deplete almost any one of our immune compartments or alter function, at least using animal models, we will see a shift in the composition of the microbiome or even the location of the microbiome or different components. And so whether that then is meaningful is, I think, kind of depends on the model, and it depends on the target. But I still think there's quite a lot to learn there. I think what we're really interested in is understanding how different microbes are driving either, health in the gut or, might set the intestinal environment up to be more predisposed to, disease pathogenesis.
All right. So then the follow-ups, what are they doing to our histones? I think that's a good place to start with. So the bugs I know that's been a big area of your work.
Yeah, I mean, our work has really focused on the epithelial cell being at the barrier. And so I can speak largely to this histone deacetylase that I've worked on for a very long time. And it's just one of many. It's called HDAC3, but it seems to be quite unique in the gut. And, I think probably many people know that HDACs like histone, this HDAC3 are inhibited by components that are made by the microbiota or metabolites made by the microbiota called short-chain fatty acids. So butyrate is one of the most potent HDAC inhibitors. We've known this probably well before we've really been thinking about the microbiome in my graduate work, which had nothing to do with the gut. We use butyrate as a positive control to make sure that HDACs were being inhibited. And so that, I think, is a real obvious connection. But I think what we're interested in is that it's unlikely that's the only connection. And so we've identified that there is at least one metabolite that can oppose butyrate and activate this histone deacetylase. And in that way, will have different effects than inhibition. But I think in reality, when we think about the microbiome and the diverse set of microbes and metabolites that are made, we're dealing with a really complex set of metabolites, and many of them are probably targeting different epigenetic regulators, either directly or indirectly, and that's going to have a very sort of profound effect on the epigenetic profile of our within our cells. And so we've focused on epithelial cells, but that's just one compartment. I mean, obviously in the gut, there's so many different types of cells, whether they're immune cells or non-hematopoietic cells, and those could all be sensing these metabolites as well.
So I think, well, of course, how these metabolites can affect the development of the immune system is so interesting. I actually was very impressed by the notion of butyrate affecting particularly Treg differentiation and activation. And I think that's what the story that you're talking about is also related to, that you do this in these mouse models. So, but maybe would you like to give some kind of mechanistic understanding also, because Jason said they modify your histone. So why would affecting histone acetylation do anything in the immune cell? So what are concrete examples that you can bring from your research, or for maybe research that you draw inspiration from, for us to understand how it goes from, food that you ingest, microbes that affect, change it, compounds that go to your epithelial cells, and then all of a sudden you have different immune cells, you have disease, or you have more disease or less disease, or...
Yeah, for sure. I'll preface this by saying this, we still have a lot to figure out. But at least one example that I can tell you is from work that our lab has done most recently. And that was looking at intestinal stem cells, and we found that butyrate would inhibit this HDAC3 in the intestinal stem cell compartment. And that actually led to what would be an increased level of acetylation at a pathway that affects the development of tuft cells. And so what we found is that by altering this histone acetylation pathway, we had a loss of tuft cells through just the impact on the stem cell compartment. And the hard part I find with thinking about epigenetics and histone modifications is we're really not able to easily in vivo modulate the mark itself. And so we are left manipulating the enzyme that creates the mark. And so it's very hard to sometimes tease apart how much the epigenetic change is really driving what we see as a phenotype. But in this case, certainly altering the histone acetylation status at this regulator of tuft cells seemed to be critical for how a stem cell could differentiate into this important lineage in type 2 immunity.
And you also see this generally with innate lymphocytes overall, right? So there seems to be a theme in your work that HDAC3 is that is modulating just general immune stemness. Is there a conserved program, or is the histone deacetylases activity different in each cell type and yet still convergent on being important?
Yeah, that's a great question. And I will say that it's mostly work by others that have looked at other cell lineages with this HDAC or other HDACs. But collectively, the takeaway, I think, is that there's some overlap in the pathways that are regulated. But what's interesting is that they're not exclusively redundant. And so inhibition of an HDAC in a macrophage is quite different has quite different of an impact than affecting an epithelial cell. That's partly to do with what their normal function is. And so whether targeting a certain pathway would have a pro-inflammatory effect or more of a protective effect. And so that is an interesting point. And I think what hasn't been done too much is really doing those cell-to-cell comparisons. But right now, instead identifying in different lineages that these HDACs are regulating both unique and overlapping pathways.
And are there examples? So you're focusing on the gut. I guess it makes sense because that's where the most of this the butyrate is. And is there examples somewhere outside of the gut that we also see the effects? Do this butyrate get in the blood and go somewhere else? Like, is there longer ranging effects of the microbiota on other immune cells?
Yeah, I think that's a really expanding field of research right now. And certainly butyrate is one of the more well-characterized metabolites that has been shown to have extraintestinal effects, whether that's in the brain, the lung. I think likely almost any tissue right now has been to some degree looked at with regards to butyrate or short-chain fatty acids as a whole. And that's being produced in the gut. And so I think the appreciation for, I'm completely biased, of course, but the fact that the gut is driving so much of what our body is doing and how, the foods we eat are impacting that, I think is from many different angles. And, I think investigators that are focused on other aspects, extraintestinal tissues, still care about the gut and how the microbiome and metabolites like butyrate could be affecting it. And I think we are quite limited in thinking that it's just butyrate, though.
I have a question. This is probably a little bit of a general question. I always wonder sometimes when, there are researchers that do some research that is in a way very applicable to like everyday life in the sense that this is something that you're ingesting, that's the microbiota that's in your intestine. Is there any knowledge that you have acquired that has impacted your habits in a meaningful way? I should start asking this to all of our guests, I think, but yeah, especially when it comes to are you now modifying your histones on purpose? Would this make sense or things like that? Is there anything that you have learned that you apply in your own life?
Yeah, I think I actually get asked this all the time. And I think that it's always in the back of my mind. I'm not going to lie. So I certainly think more about the microbiome and with regards to what I eat. And hand washing and antibiotics and decisions around that. Does it change much of what I do? Probably. I mean, probably not, because I think also, as a scientist, we are so aware of this, like how these experiments are done, that the reality of maybe one thing changing it is not we know where that fits in the context. But something like antibiotics and taking them when you might not need them is certainly something I've avoided. For my kids more recently, is certainly something that we would opt against more, unless it was necessary. And so in that way, I think it has impacted a little bit. Do I think about my histones? No, I will say no. I don't on a regular basis. I do think about fiber and short chain fatty acids though.
That's what I was going to ask. Are you up in the fiber a little bit and the butyrate just because it's good, and histones are part of that later?
Yeah, and I think, the reality is butyrate, does affect histones, but also does many other things. And I think, it's a fuel as well for the colonocytes. And I should also say that, inhibiting HDACs isn't always a good thing. And in fact, I think in the epithelial cell, there's evidence from our work and others that in the epithelial cell, it might not be very good to have lots of butyrate. The reality is, though, I think our gut can handle that, whether it's, work showing that there's limitations for how much of the butyrate gets down to the stem cell compartment. And then there's other cells that seem to, like regulatory T cells have, positive effect from it. So, but yes, I am well aware of the sort of the fiber literature, and there's some that's contradictory. And I think that is because it's we're still trying to understand all the impact of this metabolite.
So we're talking a lot about butyrate now. Are there any other particular metabolites that you have studied in your lab or that you think are particularly interesting? I think we are hearing, there's so many things. It's almost overwhelming. There are so many different chemical compounds or part of your diet. But are any of those, I think you did some work on retinoic acids, for example, that also you think are particularly important when it comes to this interface between your microbiome and your gut and everything beyond?
Yeah, I think so. For our lab, I can say the ones that we've really kind of identified as having a role in the gut are retinoic acid being produced by the certain commensal bacteria. We're very interested in trying to understand this more, because we know so much of our immune system is regulated by retinoic acid, or can be regulated by retinoic acid. And now what we're trying to understand is how much of that is retinoic acid that's being produced by our microbiota rather than from our cells themselves. So we're very interested and excited about continuing to do that. We did identify this inositol phosphate that is derived from components of our gut. I'm sorry, not of our gut, from components of our diet. And that can be generated by bacteria. And those also can influence HDACs, also, I'm sure, regulate many other pathways. I think probably what we haven't identified yet is going to be really interesting down the road too. Even though we know there's so many metabolites, we're also kind of limited to what we can identify. And there's many metabolites that are even unidentified in what they could be doing. But this concept of trying to differentiate host-derived effects versus microbial-derived metabolite effects, I think there's a lot of room there still. And so we're excited to keep doing that.
And just going along the retinoic acid thing a little more, I assume you're obviously familiar with like the Accutane and links to colitis and other things, which, common acne medication that blocks retinoic acid. I don't know if you think there's if that's like a positive crosstalk signal, right? Like if you're starting to look at the effect on the epithelium and everything you're doing, if you think that's going to link back.
It could. You mean, I think potentially from what's absorbed, what's brought in through the skin?
Or the oral Accutane associated with ulcerative colitis.
Yeah. I will say, honestly, we haven't really explored it from that perspective. What we What we are able to do is take microbes that can make retinoic acid and those that cannot, and compare their effects on, colitis or regulatory T cells, other populations that we think will be sensitive to retinoic acid, and whether in the gut they're driven to any degree by the microbiota. But I think that's a really interesting question in terms of how the retinoic regulation of retinoic acid that we ingest. The most we've done is really depleting through the diet. So really just depleting retinoic acid stores, and what we find is that then the microbes that can generate retinoic acid, if they don't have vitamin A in the diet, they don't generate it.
Hmm.
So there probably is an impact on the drugs, as with other drugs as well.
So I guess maybe to start kind of wrapping up the idea. So we have discussed, the microbiome has many potential effects on immune cells, on epithelial cells of your gut, and then this can be done through, epigenetic programs, acetylation of histones. In your opinion, or like where the field, like thinking about the future of the field, I mean, there's so much, right? The microbiome, there's so many interventions possible. There's so many metabolites you can study. What do you think kind of excites you the most about the where the field is going, and the potential for therapeutic interventions that really make a difference?
No, that's a great question. And kind of the nouns that you really focused on just there, I think all fall into this one area for us that I think is most exciting, which is thinking about how the epigenetic profile is really impacted when you first see your microbiome. And that's really early in life. So at birth, when you have colonization, what is happening to program those stem cells that could have an impact later in life? And we're getting all these clues now that, early life disruption, whether it's through antibiotics or things, can have an impact on disease development later in life. So really understanding that critical window of how the immune system is kind of instructed by what it first sees from the microbiome, and if that's epigenetically sort of coded or primed in a manner that directs the host one way or the other. I think that's a really impactful and important area in thinking about the microbiome and epigenetics together.
All right. Well, we've gone in a little personal bit with diet and how much you think about histones, but we always kind of try to wrap up these conversations with another little insight into the person behind the science. And so we like to finish with what we call the fun question. And for you, it's if you had one hobby you wanted to pursue but never been able to go after so far, if you got another 2 hours a day or 3 hours a day, what would you do?
Yeah, there's probably many, to be honest. But one thing that I've never tried, that in the era of our phones being attached to us, I've become much more interested in, is photography. And certainly would, if I had unlimited time, be excited to kind of explore doing this hobby and figuring out if I'd probably do it more from the perspective of trying to capture interesting moments, but never done it before, so who knows, maybe I wouldn't like it. Definitely has crossed my mind.
Will you also be like the photography in which you are like developing your own pictures, or just like a good old
That's
digital?
a little bit why I paused, because that part does not
Okay.
I'm too impatient probably for that part. So it has to be some mix of digital.
Got it.
For me.
Well, then I guess I'm not going to ask you to do nature photography, because you really need a lot of patience for that. Just
yeah,
sitting for 5 hours waiting for the bird to step next to your camera and not see you. So I would say that's probably not for you.
Yeah, like I said, I don't know. I could try it and actually not enjoy it, but we'll see.
Well,
that's a good one. All right. Well, thank you for coming on. I mean, I could nerd out on the microbiome for way too long, but immunology, not the bacterial podcast.
I like to be able to highlight the epithelial cell and in the Immunology Podcast.
The epithelial cell is the best immune cell.
No, don't get him started.
I didn't say
it. No.
It presents antigen. It has MHC2, and HLA, and depending on the organism, right? Like how is it not just a really awesome macrophage?
It's just a pretender. It's a pretender cell. I'm sorry. It's not, does not come from the bone marrow.
I think you would be outnumbered, Brenda.
That's okay. You know what?
They're all important.
All the cells. All the cells matter. They all make all make us working together. So I appreciate them all.
Yeah.
But the T cells, most of them. But with this anyway, no, but I think jokes apart, I think it must be so hard to like get good. I mean, mouse models are great, but then also must be really difficult when you want to make sure that this thing, how does it look in humans? I can imagine there's some differences too. So most of this work is like working mice. You can give them whatever you want them to eat. You can, make them knock out. You can get the cells from their intestines. Sometimes I wonder, how difficult it is for you to like translate to humans. I know we're kind of at the end of the conversation, but I just thought about that because it's tough, right? You cannot see the intraepithelial cells of a human subject.
Yeah, there's some limitations. I'll say though that in the last 5-10 years, we've been very fortunate to be able to develop these organoid systems that allowed us to do a lot more with primary human cells. And so we have really wonderful collaborators where we're able to obtain biopsies from patients and grow, intestinal organoids and look for, if mechanistically targeting this HDAC or giving this metabolite drives some of the pathways that we've identified in vivo. Right. And also the opposite, obtaining samples and looking for epigenetic changes in a patient sample, and then using our animal models or organoid models to test whether those changes are meaningful. So I think we've come a long way, at least for the gut, and being able to translate, and now doing co-cultures in these models has also enabled new questions to be addressed.
And thank God for colonoscopy, right?
You gotta get a little piece then?
Well, it happens all the time, right? So there is actual samples that you can get that don't require, a human to have uniquely suffered, right? It's routine medical.
They already suffered through the whole thing anyways. So you can actually donate your colonoscopy contents for science.
The stool and also biopsies and also, for where we are, there are patients with inflammatory bowel disease that are getting a routine and biopsies taken of their intestine as well. So those are also samples that are quite unique that we can translate from.
All right. Okay. Well, with that being said, with that happy note, thank you so much for joining the podcast, Dr. Theresa Alenghat from Cincinnati Children's Hospital. And it was such a pleasure talking to you today.
Yeah, it's been fun. Thanks, Jason. Thanks, Brenda.
Thank you. That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at @Immunopodcast or via email info@immunologypodcast.com with feedback or to suggest guests. See you next time.